The purpose of DNA extraction is to separate the DNA from the cell to do genetic analysis. Add 1 tablespoon of dishwasher detergent to the water. This detergent contains sodium lauryl sulfate, which removes fats and proteins from cells. Since membranes are made of fats and proteins, the membrane is dissolved and the cell pops open, allowing the DNA to come out. Then add 1/4 of a tablespoon of salt to the mixture. Mix the solution thoroughly. Using a spoon, scoop the kiwi into the plastic bag and seal it tightly. Then smash this fruit until no solid chunks are left. Add 4 tablespoons of the dishwasher solution into the bag. Mix the contents thoroughly for 2 minutes. Then place the strainer over the jar and pour the solution to filter out the liquid. Then slowly pour the rubbing alcohol until there is about 1 inch sitting above the solution. The rubbing alcohol causes the DNA to precipitate and bunch together in the alcohol layer. After sitting for 3 minutes, the DNA has bunched into this gooey white substance. Overall, this lab worked very well. I was able to extract the DNA from the kiwi successfully. I followed the exact instructions in the lab I found online, except I swapped the kiwi for the strawberries. Another difference I made was I realized after that the detergent that I used was already watered down with water, so the ratio of detergent to water in the mixture was off, but the experiment still works, so that tells us that maybe the ratio of detergent to water is not a huge factor in this lab. Another thing was that the DNA was a little bit choppy after the experiment, and a way I could have avoided that was to tilt the jar when I was pouring in the rubbing alcohol so that the alcohol doesn't penetrate the layer of the kiwi and detergent mixture. That way, all of the DNA will rise up as one piece instead of being cut.